I've got a question. Mark, right? Mark asked the question about viruses. What is the media for viruses? Yes. That's a very short answer. Cell is the media for viruses because they are obligate intracellular parasites. What does obligate means? Yeah, must. Have to. They must have a cell. They cannot do anything outside of the cell. So we grow viruses in the cell. So this is how we grow cells. I grew 3 different viruses in God knows how many, well, not hundreds, but probably maybe a dozen of different cell types. So pretty much what happens is you have cells in suspension. You put them in a plastic flask like that. And the most convenient cells stick to the surface of the plastic and grow on the surface. You can actually look at them and you see separate cells. Okay? And then you take virus, add it to the cells, and it infects them. Okay. Now, where do these cells come from? What do you think? Tissue, yeah. You can actually take practically any tissue. Add some proteases to it to break down cells one from another. Okay, break down the adherent junctions, adhesion proteins, and just put them on and leave them and hope for the best. Some of them will stick, some of them will not. You may end up with a culture. This is called primary culture. You can just, seriously, you can take fat and grow culture of fat cells. Okay. The only problem with cells taken from the tissue is that so you can grow them, they will occupy the whole surface of the flask. And at some point, they will stop growing because all surface is taken. You can collect them, again, treating them with a chemical, with an enzyme like a trypsin to break down the attachments, okay? And then you can put them in the new flask, the bigger one, and they will grow. What's the problem with the cells coming from the tissue? What cells normally do after some time? They die, yeah. So they're going to die. But every time you change the flask, it is called a passage. After 4 or 5 passages, they're going to die. Which cells don't die? Huh? So, how can we cause mutation? There are some, yeah. You can add, there are some cells that have part of the virus in the genome any other way. How? Temperature doesn't cause mutations in mammalian cells, not that much. Yeah, you can UV light them, you know. Everything that doesn't kill us makes us stronger. Well, in this case, it makes them mutant. Okay, the ones that will survive will probably become essentially cancerous cells. They will grow forever. And that's convenient. The only problem is that they're slightly different from normal. So you have to keep it in mind when you grow virus in the cells and you observe different viral properties. A little digression. Have you ever heard about, yes?
Does UV light kill viruses?
Oh, you do it before viruses, you know? So you collect cells, you irradiate them, and then the ones that survived can now grow forever. And you can use them to pass the cells. Does that make sense? To grow virus. You can. Have you heard about Henrietta Lacks? Yep. Good. Henrietta Lacks was an African-American woman in mid-50s. She was diagnosed with a cervical cancer. And eventually she died from cervical cancer. But at some point, her tumor was excised and was used to make a cell culture. Actually, cell culture was invented in, I think, like mid-40s by Renato Dulbecco. it's not that old invention. It's like 20th century. Anyway, our cells were used just for the lab purposes. They grew poliovirus on it. They grew influenza. It's probably easier to say which virus they didn't grow on HeLa cells, called HeLa cells. Why there's so much buzz about Henrietta Lacks? Actually, there's a book called The Eternal Life of Henrietta Lacks, or The Immortal Life of Henrietta Lacks. Because HeLa cells are used everywhere in the world. Seriously, maybe 1/3 of all molecular biology is done on HeLa cells. What's the problem? What do you think? They very much different. Actually, if you will inject it into the mouse will develop a tumor right away. Human probably too. What is the ethical problem? Any idea? Yeah, they didn't have. Now, this is a very tricky and slippery slope that I'm stepping on. At the time they collected the specimen, in like 56, there was no consent. No such thing. Nobody asked you because nobody thought about it. You don't need that tumor anyway, we're going to use it. Okay, now we have to ask for consent. If you give a part of your tissue, if you give your blood, in some cases, doctor can ask you, would you sign the consent form? And it's your right to say, no, I don't want my blood to be a part of any medical research. Now, what is the main implication of not having consent? What do you think? Do we know the human genome? Pretty much, yeah. We sequenced it. What about the specific genome of those HeLa cells? We know, yeah. The whole genome of HeLa cells was sequenced. Now, do you think Henrietta Lacks had any relatives, descendants? Oh yeah, a bunch of them. So now we know the genetic makeup of those people without their consent. You see that? You see the problem here? And we now enter the century, the time when genetic information is not something from sci-fi movies or futuristic forecasts, It's going to be a new reality of healthcare, and we'll chat about it a little bit. Okay, but that's kind of an interesting issue. And from one standpoint, outrage of Henrietta Lacks' family can be understood. She was never asked if she gives the permission. But on the other hand, there was no law about it. Doctors didn't even think about that. Okay, so that's kind of a tricky part. I gave my cells blood cells for research purposes, but I wasn't identified. So, I mean, if you read the paper, it doesn't say, these monocytes were derived from Dr. K's blood, thanks to him. Okay? No, it's all blinded, but nevertheless, now you have to sign a consent form. Now, cells, in turn, have to be maintained in the medium. Of course, you don't have to know the components. It's a defined medium, but you can quickly make it complex because cells often require the serum from calves, fetal bovine serum. When calves get butchered, should I say that? Butchered? Okay, when they get butchered, they collect the blood, they isolate the serum, and that serum is used for cell growth purposes. It contains growth factors that cells need. Okay, so you immediately turn it into the complex. So we can't really say if the medium is complex or defined for viruses. All we can say it's cells. Okay. We can use live organisms as a medium. We can use eggs. We can use rodents. Okay. Eggs are widely used for growing influenza virus. there was an interesting, and actually the influenza virus that is used in vaccine preparation is grown in the cells. You can actually see the person inoculating eggs with influenza. It's kind of a boring job, I would say. Really boring job. Now, there was a funny story when a couple of years ago, influenza vaccine production was under a threat of the egg deficit. There was egg shortage in the United States. So they had to buy, eggs became more expensive, so they had to jack up the prices for the eggs and buy more high prices. You know what was the reason for the shortage? Bird influenza that killed all the chickens. So in order to get influenza vaccine, we have to get the eggs from chickens, but chickens got killed by their own flu, against which we don't have a vaccine. It was kind of ironic, in my opinion. For some parasites, like worms, filarial parasites, you have to use rodents to actually grow the parasite. You can't use cells, so you gotta use animal. Question? Now, we can all understand why we need actual animal to grow a worm. But we have those wonderful cells right here. Why do we still need eggs? Why do we still grow vaccine in eggs? Why don't we just switch to the cells? Oh, cells much cheaper, way cheaper, and they occupy so little space. It has nothing to do with science. What do you have to do when you change a major component of the drug? Huh? Say again? Oh, well, you can patent it before. Yes, you have to get it tested and approved, and it costs a fortune. Okay, so instead of going through all those hoops with FDA, they will just stick with the eggs. Just easy. Now, what disadvantage of the eggs and why they were used? Well, why they were used in the first place? We didn't have cells, and they were cheap. And they are cheap. Now, are they the same eggs that you buy in a grocery store? What's the difference? Not really. Not necessarily, no. They have to be pathogen-free. They have to be certified as specific pathogen-free. So they must have no possible viruses, bacteria, and so on and so forth. So they're a little bit more expensive. Okay. Even plants. A lot of virus work, like basic work, is about studying the plant viruses. So this is a leaf of a tobacco plant infected with tobacco mosaic virus. And to be honest, the names for plant viruses are the best names in the scientific world. They're really creative. I don't remember all of them, but my favorite is the bushy stunt tomato virus. Well, guess what it does? It creates tomatoes that are bushy and have stunted growth. So, and trust me, it may sound silly. USDA pays a good buck to people who study those viruses because they are important pathogen of crops. So that's, at some point, I was working in the University of Nebraska, and we didn't have money. We were not really well funded. And people in plant virology were laughing at us because they had a ton of money from companies, from USDA. They were funded really well because they were studying something that can affect food supply. And we were studying some stuff. Now, once you get inoculated, once media get inoculated, you get it ready, you stick it in the autoclave. Why? Why autoclave? Why don't you just leave it here? Controlled temperature, very good. Controlled, it's controlled temperature, it's controlled environment. You can control temperature, atmospheric composition, humidity, everything, okay? You can adjust the growth conditions. Why it is so important to have conditions controlled? Which means, yes. So if you grow it later. You can grow it later based on your lab report, okay? That's why controlled conditions. They allow you to replicate, okay? They also allow you to control for certain parameters. Say, if you, now look, if I place it here, I have no idea what the humidity, temperature, and stuff like that. So if I change pH of the medium and they don't grow, I don't really know if it was because of the pH or the draft or something else, okay? But if I stick them in the incubator, I know that environment is going to be exactly the same as it was last time. Does that make sense? Growth can take days, weeks. We grow it for 2 days usually. That's why I prefer to do the labs on Tuesday. So by Thursday we can see the results. Technically, we're gonna see plaques tomorrow. We can't see them tomorrow, but by Thursday they're larger. Okay. Some microbes are not for the impatient. Bless you. Mycobacterium tuberculosis may take up to 2 weeks for the first colonies to arise. It's awful. I wouldn't be able to work with it. Yes.
Is there any instance where going from collecting the microbes and then going to the incubator is between that time period? Is there any way of it impacting the test results?
Excellent question. So you collect, say,
because they're not in the incubator.
Yes. So you go from cave to your lab. Can they die? The brief answer is absolutely they can. There's a possibility. So this is something you have to think through. Yes. In some cases you need transport medium. Say you collect presumably anaerobic microbes. You can't just expose them to the air. You have to stick them in some sort of a container that would preserve them. Does that make sense? You collect some photosensitive microbes. Same. Some viruses are, if they are taken out of the organism, they may start dying at the normal temperatures. You have to freeze it. So you have to think it through. That's a good question. Okay? So it may, when you report your test results, you have to report how you transported it. Does that make sense? This, from here to there, not really, no. So if I will leave this thing on the bench right there, we would have some colonies by Thursday. They won't be so well developed, but we would have some. Does that make sense? Now, colonies grow. The environment, the colonies that you've got from your hand washing experience, again, going back to it, I have several types, right? Say you're really curious about that yellow stuff. That's what you got to do with it. You got to isolate it. So that's why we use agar, solid medium. So you can pick one colony, get a little bit bacteria from it, and subculture it. Does that make sense? You can subculture. You can put it on the agar plate, or you can put it in the liquid medium and grow a bunch of microbes like this. You see? That's the liquid medium with serratia in it. It grew in that medium. What would be your reason to go either way? Rationalize it for me. Why would you have a yellow colony and you decide to put it in the liquid medium? Why? To grow it. right? To just accumulate it. What would be the rationale to put it on the agar plate again? Expand. In terms of colonies, yes. But what? Yes. Good. So colony formation without competition. Any other reason? You don't take into account the human factor. What if your colony that you think is just yellow, what if it's mixed? You want to make sure that it's really only yellow. We had several times before somebody has clearly just one colony, picks it up, does the subculture, and it's like 3 different species come out because 2 of them were hidden. Okay, so that's one of the reasons to maybe go 1 step ahead with the subculture, because once you get this culture and you have 2 species in it, you won't be able to separate them. Does that make sense? You can isolate colonies only on a solid medium. You want to have be 100% sure that you have 1 species before you inoculate the liquid medium. Make sense? So now colonies have to be separated. Why we focus so much on the colony? It's pretty much a reason why. All cells are presumed to be the same. So when we isolate a colony, we suggest that we isolate not only species, we isolate a very particular strain, if you want, of microbes that is unique, because maybe in next colony there are a couple of mutations. Okay? So here you can see, that's the mixed culture. We put it on the plate and they get separated. Okay? And then you can, in ideal world, it works like this. In not ideal world, it works like this. Okay? So you can see how all the different species are mixed here. And here you can see E. coli, Micrococcus luteus, and Serratia marchensis. How can you achieve that? How can you make colonies farther apart? It's simply a method of technique. It is called a streak plate. Now, the easiest way probably would be to open the notebook, and I will just try to picture it for you. This thing will, yeah. So you take a plate. I'm going to use this as a plate. And you take a loop. And you collect a colony. Not even pick, just touch. Okay? Or you take a sample. Okay? Of whatever you sampling. Okay, so this is your plate. So what you do first, you take your loop with whatever sample you have on it, and you do this. Is it going to be dense in terms of bacterial growth? Probably so, because you have a lot of stuff on the loop. Next thing, you take your loop and you place it into the flame. What happens to everything on the loop? It dies. So it's sterile now. Take sterile loop, not touching anything, just thin this out. Does that make sense? you collect whatever bacteria you have in this area and spread them along larger surface. Does that make sense? So density-wise, this and this, which one is going to be less dense? The right one, right? Then you sterilize it again and you spread it again. You start by picking something from here. Does that make sense? Okay? You make it even less dense. And then you sterilize again and you do this. Okay? So you thin it out. Alright? For better guidance, to make it even easier, when we're going to do the strict plate isolation, you can do this. This is the plate. You can separate it like this. So this is your first round of inoculation. It's going to be very, very dense. Alright? Then you do this. It's going to be thinner, and then you do this. It's going to be even thinner. So you consequently thin out your bacterial population. And you will see how amazingly it works. When you start with a mixed culture and you end up with large colonies clearly separated from one another. Does that make sense? Yes.
With this method, do you risk using some of the microbes that are
Absolutely, yes. If some microbes, if you have one, two colonies in your sample and you don't grab them, then you may lose them. They may not appear on that blue one, okay? So some of the colonies that were present here may not show up here. Does that make sense? If they're just not very abundant. Is that good? Question? No? You good? Okay, so that's one of the very, fundamental methods of isolation. That's really, again, some things I get really excited and I can't really stress out how important it is. This is like turning steering wheel when you drive the car, okay? Nobody wants you to do the drift, when you just learn how to drive. But you better turn that damn steering wheel, okay? So that's foundational. Other ways to thin it out. Loop dilution. We will do modification of that method. So in the loop dilution, you take microbe and put it in the liquefied agar. It has to be just warm. It shouldn't be hot. It will kill microbe, right? But it should be liquid, just warm enough to stay liquid. You add it to the agar and you mix it really well. You sterilize the loop and you transfer some of this diluted sample into the next tube. That makes sense? So you dilute it even further. And then you transfer it to here, even further. Right? Follow me? So you subsequently dilute your sample. And then, after you prepare each sample, you pour it in the plate. And the number of colonies decreases. This method, if a little bit modified, allows you to count the number of cells in your sample. You can just take the exact volume of the liquid culture. Say you take 100 microliters, okay? and you dilute it in 10. So you have 100 times dilution. And you do 3, 4 consequent 100 times dilutions, you count number of colonies, and you have a pretty good idea what you had in the beginning. Because you know the volume that you've taken, and you know how much time you diluted it. We're going to do that as well. Spread plate is the simplest. Use the hockey stick. That's hockey stick. So take a sample either with a pipette or with a wire loop. Place it in the center of your media, center of the plate, right here. Take hockey stick and just spread it well and evenly. And that's it. It is often used to prepare so-called bacterial lawns, when bacteria occupies the whole surface of the plate. We will do it when we will test the effectiveness, bless you, the effectiveness of different antibiotics. That's actually also pretty fun. You just spread it, okay? So, imagine now that you have selected the colony. So you subcultured it, it grows in the liquid medium. Now you have to figure out what is the bug. So first you inspect. That's what we did on last Thursday. We identify the shape of the colony. They were all round shaped with the clear edges. Well, some of them were not so clear edges. Identify the color. We identified if they're motile or non-motile and stuff like that. We actually put them under the microscope and looked at them and identified the shape of the cells. And in some cases, we were able to say that staphylococci exist in the form of single cells, while micrococcus exists in the form of double and quadruple groups. Okay, that makes sense? So we're able to give some characteristics. We could stain them, look at them, you know, under the gram staining. We could do a lot of stuff. Very basic techniques, and they would help us to narrow down what those microbes are. That makes sense? That's your inspection. Let's put it this way. Now, when we say identification, what we mean? In this case, we want to know exactly what it is. Why? Good. That's actually a very good answer. Yeah, and treatment. Really. Treatment. Treatment's not knowing what it is, you won't be able to decide on the best treatment, right? There are a lot of various tests, okay? You can do some biochemistry. Remember we talked about, I don't want to go all the way back to that slide, but remember the medium that becomes black if microbe reduces sulfur? That's biochemical test. So it possesses some enzymes that reduce sulfur, and we can detect it by that it forms iron sulfide. We can do immunology. How many of you are familiar with the concept of antibodies? Okay, those who aren't, I'll briefly explain what they do. Okay, you can check DNA, and we'll chat about it. So there are a lot of various methods. Okay, and I will introduce you to some of them. And before we get any further, any questions on the techniques and separation techniques, isolation techniques? More or less clear? Good. Methods. So I want to give you a little preface to this little chapter. For a long time, microbiology textbooks and microbiology classes, I think, were omitting the current methods that are used to identify microbes. Probably because these methods are complex. And if you ask me, they are complex. Does that mean that you shouldn't know them? In my opinion, absolutely not. Because I think nothing is more wrong than you coming in the clinic or whatever job you're going to take and talking to a person and realizing that you have no freaking clue what this person is talking about. That you are not understanding. I'm not trying to teach you the protocol so you walk out of this class knowing how to do with your hands everything. But I really want you to have basic conceptual understanding of these methods, because you may, and many of you will, run into those methods once you start working in the clinic. If you plan to become, does anyone go into the biotechnology? Okay. Yeah. Dr. Deek will write you about those methods. Okay, just for the sake of the classes. Okay. You start working in the lab, you're going to use a good half of them. Those of you who go into the healthcare, I will tell you which ones are going to be used in the clinical lab. And you're going to interact with the clinical lab, unless you work in radiology. They're not really interested in microbes. Anyway, tell me if you don't understand something. Okay? I want you to have a conceptual, basic understanding of what's going on. So first are immunological methods. All those methods are based on the idea of antibody recognizing the antigen. And we will insert, well, blank page. Okay. So we're going to chat. Yes. We'll get to that. We'll get to that. So I want to explain the underlying concept in the very first place. We have immune system, and mammals have immune system, right? So when we get something that's not ours, something that is called antigen, okay, our immune system responds to that something and produces antibody. Any questions on that? This is what your flu shot is based on. All shots are based on that response. You put some antigen into your cell, you produce antibodies. If anybody has peanut allergy, your peanut allergy is because of the antibodies that your body produces against proteins that can be found in peanuts. Okay? Is that clear? Now, very, very, very important feature of that interaction is that it's specific. Which means, if you have an antigen that comes from influenza virus or Staphylococcus aureus, antibodies to influenza will never recognize Staphylococcus and vice versa. Moreover, antibodies against proteins of Staph aureus will not recognize Staph epidermidis. Does that make sense? So they are perfect specific tag for a strain, even a strain. You see? You can very, very specifically recognize it. What can serve as the antigen? We're going to focus on proteins mostly. Okay, proteins are perfect antigens. Any questions about this part? They probably will be. But now let's talk how it works. First, we're going to talk about Western blot. Microbes consist of what biological macromolecule the most? Macromolecule. Which polymer, biological polymer, is the biggest structural part of the cell? It's an organelle. What is the biggest molecule? Which molecule carries the most structural functions in the cell? Molecule. Protein. It's protein, right? So a lot of protein, and there are a bunch of proteins, different proteins. Okay? So you can take the cell and break it in pieces, and it's going to be just a bunch of proteins. Like you can take a human and chop it in pieces, and you can have ears on one side and hands on another. Okay? Now, proteins are of different size. Okay? You put those proteins in what is called gel. And trust me, it looks like a gel, and it walks like a gel, and it walks like a gel. Proteins are charged. So apply electric current, and proteins start to move. In that solution, proteins are negatively charged, so they move to the positive end of the gel. Does that make sense? Now remember, I told you they're different size. Which ones are going to be moving faster? The smaller ones, yeah. Smaller are faster. So you can separate them based on their size. See that? Gel is awfully inconvenient to work with. It can break down. You pretty much just can separate it. So what you do next, you take a gel and take a special paper, a nitrocellulose paper. Well, it's not a paper. We call it a paper, okay? And you, again, using electricity, you transfer proteins from the gel onto the paper. You put them together in a special machine, and proteins just move to the paper. Now, paper, well, that little sheet of nitrocellulose is very easy to work with. So throw some antibodies to that sheet. Antibodies stick, but they stick not everywhere. They stick only to the protein they can recognize. Does that make sense? Does that make sense? So, if you have that protein, you're going to see the band. If you don't have the protein, or like you see the band. If you don't have the protein, you don't see the band. Okay? What do you think are advantages of this method? Bingo! You can very specifically address one protein. So if you have two strains that are different by one protein, you can distinguish them. Yes? Good. So it shows you the protein that you look for, or doesn't show, but it doesn't show how much of that protein you have. Make sense? Was that your question or not? You can ask it again. Yes. Say I want to check for, I don't know, lactase in E. coli. So I have antibodies that can recognize only lactase. Right? I add it to the paper. If there is lactase, they will stick. If there is none, tough luck, you know. I won't see anything. Does that make sense? Oh, it doesn't. Paper is just a carrier. Proteins will stick to it. So if there is a protein in a sample, I'll see it. If there is none, I won't. So it allows you to recognize specific protein. It is awfully sensitive. It can detect 10 to the power of negative 12 grams. It's freakishly, absolutely just, current methods allow you to just see the minute amounts, okay? Downside, it's not very quantitative. Not exact. You can't say the exact amount. It's pain in the ass to do that. And if you look, you will find only what you're looking for. Does that make sense? If there is something that you're not looking for but interesting, you're not going to see that. And it takes an awful lot of time. If you start at 8 a.m., you're done by about 6. Yes?
So is this how a titer is done?
Not really. Titer is when you do the serial dilutions of something, of antibodies, okay? And see which dilution exerts certain effect. Has certain effect. Does that make sense? Or not?
No.
Okay. So I like, now take Mark as an example. I want to hear that. I don't want to see that. Okay? If you don't understand, tell me. No, I have no idea what you're talking about. You can take, say, you want to check the titer of antibodies in the blood. That's what they do in the clinic. Right? So that first you have to separate cells in the blood from serum. So you take serum. Then you take serial dilutions of the serum. So you dilute it 2 times, then you dilute it 2. So you have 2, 4, 8, 16, 32, 64, and so on and so forth, times dilutions of your serum. So antibodies that are presumably in your serum become more and more diluted. Does that make sense? Then, say you want to see if you have antibodies that are effective against, what virus do you want to take? Huh? Hepatitis B. So, hepatitis B is probably not very convenient to operate as a whole virus. So you take a surface antigen, just the protein, okay? And you have a standard concentration. Now you take the whole serum, take hepatitis B, and you have complexes, and there are ways to detect those complexes. You pretty much can stain for them, like stain the gel directly, and you see there's a complex. You take 2x dilution. Again, you have enough antibodies to form the complex. 4x, enough. 8, enough. 16, enough. 32, enough. 64, not enough. At 64 times dilution, you see no complexes forming. It means that your titer is 32. Does that make sense? That's the titer.
I still get another question, I guess.
You'll ask. Now, how do you say is that a good titer, is that a bad? It's not. There are guidelines. Say if you have a titer more than that or less than that, then you good or bad. For diseases, it's going to be if you have titer like 8, dude, you are sick or you are sick. If you have titer 512, you're okay. Next question.
So, what if there's, well, okay, so if there's no response then? What if there's, I mean, it's like if you're trying to figure out, okay, are you resistant to virus?
Okay, hepatitis B, yes.
Hepatitis B. I understand what you're using, the quantity of the antibodies as how resistant you are to getting it, right? But what if you don't have any antibodies?
you're not gonna have any binding. You're just not gonna have any complexes. Does that make sense?
Yeah. So do you apply the hepatitis B virus to the serum to determine
A part of it, a protein
a protein?
of hepatitis B. So, say you have one person that got immunized against Hep B. Okay? So this person is going to have antibodies. Another person that not immunized, no antibodies, right? So add hepatitis B, well, not the whole virus, but usually the surface antigen. Antibody will bind it. Okay? You add protein here, nothing binds it. Then you have techniques, more or less sophisticated, that allow you to detect this complex. Nothing to detect, right? You don't see the surface antigen by itself. Nothing to detect. Does that make sense? So you're going to have, in a person with antibodies, you're going to have a signal. In a person without, you don't. Yeah.
What you were talking about was basically counting
Now,
what you detected.
yeah, you can expand on it and make it semi-quantitative. Does that make sense? Okay. So that was your Western blot. Now, ELISA. This Western blot in clinical lab is not a common feature. ELISA is. It's to a large extent automated now. Because you have plates with many little wells that you work with. And trust me, pipetting 96 wells is a lousy job. It's boring. Okay, the only upside is that while it incubates, you can have lunch. Other than that, it's pretty boring as say. So we have it automated now. What it does, on a very basic level, let's say you want to find the antigen. You get the sample, you add it to the plate, and you wait for the antigen to stick to the surface of the plate. Surfaces are treated in a special way. They're like kind of sticky for proteins. Okay? That makes sense? So you have antigen sticking. You add an antibody that will specifically recognize the antigen you are looking for, and that antibody has enzyme attached to it. This enzyme will Question? Oh, this enzyme will produce some sort of a signal. It can be changing color, it can be fluorescence, luminescence, something that you can't detect. Okay? You can do different variants. This is called direct, this is called indirect, when you have to add second antibody that will recognize first. You can get antibodies attached to the plate, and that's what you're going to find in the clinic. Like when you say you want to test for hormone, like leptin in humans. Okay, you call a company, they send you a plate with antibodies against leptin on it already, and they send you this detection system. All you need to do, take serum, put it on the plate, add detection antibodies. There you go. Does that make sense? Okay, last thing, sorry I hold you a little bit back. Immunofluorescence, basically the same stuff. You have a cell with antigens on its surface. In some methods you can detect actually antigens that are inside the cell. But basically you add antibodies that can fluoresce. Antibodies stick to the antigen You turn on UV on your microscope, and you see the cells. This image is from a good friend of mine. Okay? You can see here, so neurons are red, astrocytes are green, and nuclei of the cells are blue. So you can have 3 different colors that show 3 different structures, all at the same time in the same picture. Okay? So we're going to stop at that. I will see you on Thursday. On Thursday, we will have a quiz on the studying microbes. Okay?